All right, welcome back. This is the second video, which is on the immune system. So what we're doing here is we're focusing on only the third line of defense. The first and second line of defense were covered in the first video. The third line of defense is the final line of defense that animals have against, well, that many animals have against pathogens. This third line of defense is the only one that is specific immunity, which means that it is going to be particular to very specific pathogens. It's also called acquired immunity because it's not something that you're born with, it's something that you have to develop during your lifetime, which is also why it's called adaptive immunity. All right, so the cells that are involved in this final immune response are going to be some of the lymphocytes. Also white blood cells, we also talked about eosinophils and neutrophils and macrophages and stuff with the second line of defense. And they're connected, but this third line of defense is the only one that's going to use these lymphocytes, the B lymphocytes, and then the T lymphocytes. And there's a couple different kinds of T lymphocytes. There's going to be helper T cells, and then there's going to be cytotoxic T cells that we're going to be talking about. All right, so because there's B cells, they have a really very different job. They make antibodies. That's a very different immune response than the T cells. And so there's kind of a fork in the road here, kind of a two-pronged, I guess, attack in the third line of defense. And they're kind of separate from one another. All right, so the B cells are going to be involved with what's called humoral immune response, which is going to be involved with the production of antibodies in the blood. And the T cells are going to be involved with cell-mediated immune response. Those particular T cells, the cytotoxic ones eventually, they do end up going to the battlefront. They go to the place where the pathogen is, and they actually cause cells to die in that area. All right, so what these are acting on, because the reason that they're specific is because they're going to be acting on a very specific component of a molecule, okay, a foreign molecule that gives this immune response. It's usually a protein. It's usually some little bit from the capsid of a virus or some little spike from a bacterial cell. It's usually some little protein that the body responds to. All right, so because that humoral immune response is going to be involved with antibodies and they act against antigens, let's take a closer look at the structure of an antibody. So these antibodies are produced by B cells. They are proteins themselves. You have genes in your genome that code for the various components of these antibodies. Antibodies are a protein that always has kind of this, well, it's got quaternary structure, and that involves some components which are constant and some parts that are very variable. So the C areas here, those are going to be the constant regions. They're always, structurally the same. Notice they're held with disulfide bridges. That was one of the things that could hold tertiary or quaternary structure together. And then you have these variable regions. Each antibody has multiple variable regions. This one has 2, but there are some that can have more than that. And what they basically do is they're going to clump together by holding on to them and binding to them various different antigens. And so here you can see the interaction here. At this spot here, you can see the interaction between an antibody and the antigen itself. This is a lock and key type fit, and that's why this is very specific. These antigen binding sites are extremely particular. There are a variety of different kinds of antibodies. As I mentioned, they're not all just that Y shape. However, that is the one that we're going to focus mostly on. The most abundant of the typical antibodies is an IgG antibody. This IgG type is the type that we usually think about when we're going to be dealing with immunity. So if you read through this, it's the most abundant circulating antibody. It does cross blood vessels, enter tissue fluids. It also crosses the placenta and confers passive immunity on the fetus, protects against bacteria, viruses, toxins in the blood and lymph, and triggers action of the complement system. Okay, so that's your main antibody. If you're interested in knowing about allergies a little bit more, you should read up on IgE antibodies. Those are ones that tend to be the problem when people have allergies. And then there's a few other different kinds of antibodies that we're just not going to be getting into here for this. Okay, I think I've gone through this. They have variable binding sites, antigen specific. Okay, so now let's get into the actual steps of the specific immune response. All right, so it does start out with a normal second line of defense piece, okay, and that's what we're looking at here. We have a macrophage, that second line of defense, and it's engulfing a bacterium. Now, they don't just eat it up and that's it, but they also kind of show off when they do that. They have these molecules on the outsides of them, which are they're proteins on the outside. They're called class 2 MHC molecules, where they will then put a spike from the virus, some component from the outside of the virus, or sorry, this is a bacteria we're talking about, but they do the same with a virus. So some component from the cell membrane of the bacterial cell or something. And they hold it on the outside. I kind of say that this is sort of like in battle when they would, old-time wars, you know, put the heads of the leaders on a stake to kind of show off. So yeah, they're showing off. Look what I caught. All right, so that's second line of defense stuff. All right, but now we're going to start to get into, and this is the connection to the third line of defense, because here we're going to start to use some helper T cells. Okay, so this is a helper T cell. Notice that it is docking up with that particular macrophage using its receptors. Okay, so a helper T cell is kind of the go-between for both of the two different parts of the third line of defense. It is the middleman. What it's going to end up then doing once it finds and receives the information that, hey, we've got this antigen fragment, is a few different things. First of all, it releases an interleukin molecule. Interleukins are chemical signals that are going to cause a lot of things. First of all, they're what cause a fever. So that signal then gets sent to the body, and it just basically makes you feel cruddy. Gives you a fever, gives you achiness, gives you a headache. All of that is your own immune system. It's this interleukin that's being produced. And then the point of it, and notice this is a positive feedback loop. We don't see those real often, but a positive feedback loop means then that this interleukin is triggering more production of interleukin, which is triggering more production of interleukin, snowball effect. All right. So this interleukin then is going to cause some of the other lymphocytes to be activated in the area. But it has to find really the correct ones. It has to find the ones that have the same receptor types that would match up to that particular antigen. So this third line of defense sometimes takes some time. But notice that it is the go-between for both the cell-mediated immune response with the cytotoxic T cells and for the humoral immune response using the B cells making antibodies. All right, so mentioned this already, that the helper T cell is going to bind to the antigen fragment on the cell's surface. It's activated by interleukin 1 from the macrophage, and then it makes interleukin 2. You read through this yourself. You went through this already, but you can review that. Okay, now let's take a look at each one of the two final responses together. So this is cell-mediated immune response. This would be the role of cytotoxic T cells. So what they're going to end up doing is they're going to end up then, once they get activated, going to the battlefront. Okay, so this would be then that they're circulating in your bloodstream until they get to some of these cells of your body that are infected. So if this is the flu, they're going to some of the cells in your trachea, for example. They bind to some of these cells that also have this same molecule on the outside of it. Basically, this infected cell is also holding out little pieces of the bacterium going, I'm sick. So unfortunately, I guess for that cell's a goner because the cytotoxic T cell binds to it, puts pores in it with a protein called perforin, which causes water and ions to rush in, odor, and then that cell explodes. Okay, so it causes cell lysis for the infected cells. We can't let those infected cells continue on because particularly if they're infected with viruses, they're just going to make more viruses. So we got to kill them all. So these are the steps here. Secretes perforin. Okay, the other branch of the third line of defense is called humoral immunity. I'm going to go through the steps here with the image, and then we can come back to those pictures or the words. All right, so what's going on here? This backs us all the way back up to second line of defense here again with the macrophage eating the bacterial cell, and then once again we're activating the helper T cells that's secreting interleukin-2, but finally this activated helper T cell, and this can take some time, is going to find the B cell that has the correct receptor. That B cell then will be cloned to make memory cells. Those will patrol your body for the rest of your life. And they will be cloned to make what are called plasma cells, which are just basically activated B cells that secrete antibodies. So the B cells never go to the battlefront themselves. They secrete antibodies, and those antibodies are proteins circulating in the bloodstream. They end up at the battlefront. So once those antibodies get into the bloodstream, once they get to the site of the infection, they're going to work in all of these different ways. They can neutralize the bacterial virus, keep it from being able to invade any more cells. They clump them together. That's called agglutination. If you're dealing with a soluble antigen, something maybe like snake bite venom or something like that, it can cause it to precipitate out, the particular chemical to precipitate out, and all of these then make phagocytosis happen more easily. It also can activate complement, which can then cause lesions in the foreign cell itself. So this is a really nice summary. You guys have this on your reading guide so that you can map out what's going on in the third line of defense. All right, active versus passive immunity. Active immunity, both of these are third line of defense pieces, but active immunity is going to use antibodies that you make yourself. So like if you have a cold like I do right now, I'm producing antibodies, that's active immunity. Passive immunity you get from somewhere else. So passive immunity, mostly, I don't pick up much passive immunity anymore. You mostly get it from breastfeeding or emergency treatment. Some particular things, rabies, measles, snake bites, what they're actually going to give you in these emergency treatments are those antibodies. So emergency treatment of those things, that doesn't give you permanent protection against something like snake bites because you don't end up with the memory cells. You're just getting antibodies. Passive immunity is temporary. No memory cells are made. All right, so if we take a look at maybe now the second time that you get exposed to the same virus, for example, what you're going to end up is with your response being very, very different. You're going to get a faster response. It's going to be more prolonged, and you're never even going to get to the point where you feel sick because your body's going to produce those antibodies so fast. Primary response can take a while. It can take days before that helper T cell finds the correct B cells inside of toxic T cells. It can take 10 to 17 days. So, if you get the flu, you can be sick for a week at least. Secondary response is quick. It wipes it out real fast, and you never even actually know that your body is dealing with it. But it is. It's dealing with this all the time. So this image here is, I think, great, showing the impact of primary versus secondary response. So here you have moment of the first time you encounter that particular flu bug for this particular year. Notice it takes a while here before you produce any antibodies. That's going to be the period of time where that helper T cell is trying to find the right B cell, and it takes a while. It's called clonal selection. Once it finds the right B cell, then that B cell starts to make antibodies, and you end up stepping up your immune response, and you wipe the thing out. So, at this time here, now you're taking care of it. If you were exposed to the same thing again, which is very likely, you're dealing with the flu bug, other people end up getting the flu bug a few weeks after you might be family, you will likely have secondary exposure to the same thing. But because you have memory cells, you don't have to go through clonal selection, and you end up producing this many antibodies very fast, and you never even know that you're sick. This is why vaccines work. It's because the vaccine serves as the first exposure to the antigen. You end up with this response to it to the point where you do end up getting memory cells. And then if you came across the real thing later in your life, then you wouldn't end up actually feeling sick because your immune system would take care of it right away. Okay, I'm not going to spend real much time on this, but I wanted you guys to have this slide so that you could study it a little bit yourself. I did want to mention HIV. HIV is, of course, a virus. The problem with HIV is that it infects specifically, because remember that viruses are very specific to their host cells, but HIV infects the T cells, particularly the helper T cells. So what ends up happening as it progresses, that you end up having your T cell concentration be depleted, so that you can't, it's like a conundrum. You can't fight it off because it's actually killing the cells that would fight it off. So if you take a look at what ends up happening over the course of an HIV infection that leads to AIDS, you can see what ends up happening with the levels of antibodies initially, but then how they end up dropping off as the T cell concentration drops off, because the HIV concentration goes up after a period of time. And these years after infection, this slide is something that's probably 10 years old, so this is probably better now than it was in the past for many people because of advancements in medicine. It's still obviously a deadly disease, but in many cases we push back the period of time before the individual gets full-blown AIDS further than what they're showing in this slide today, but not always. So that's the issue with HIV that it's important, I think, that you know that what it's dealing with, the cells that it's infecting are those helper T cells, and so that's why it causes such a horrible, horrible problem. All right, so that's it for the second video, a little bit longer than the first, told you. If you have any questions, let me know. I also do have study guides for this section that I'll have available to you guys after break. Enjoy your break.